So hello and welcome to the What Influences Enzyme Activity Lab Part 1. Today we're going to be looking at how reaction duration will influence the reaction completion, as we'll develop a character profile for our enzyme here, invertase, which is a type of sucrase enzyme that's going to break down our sucrose. So what we're going to start off first is labeling our microcentrifuge tubes, which are these small tubes here. So as always, labeling our glassware is always important, but we also need to label any of our essentially plasticware that we'll be using in our experiments, because today our reaction vessels will be these microcentrifuge tubes. So I've already gotten out the 8 microcentrifuge tubes that I'll be needing and separated them to make my labeling easier. I know that I have 5 test tubes that I'll be using and 3 control tubes. So I'm going to take the tube, and then on the top of the tube, so it opens and closes this way. So I want to make sure that when I look at the tube like this, or from your perspective it would look like this, that I can read the label and that it's not upside down. So I'll write a big capital T and the number 1 to clearly illustrate that this is going to be the test 1 tube. Now I'm going to place it slightly away from where it was so that I can keep track of my labeling as I'm going through. So now I'm going to do this for the remainder of my microcentrifuge tubes. When labeling these tubes, the handwriting doesn't need to be perfect, as we're going to be throwing these away at the end of the experiment. If we were keeping these tubes, we would also label them here on the sides and provide more information, including who made the tube, what's in the tube, and when the tube was made. But again, since we're only going to be using these for today's experiments, we don't need to. So now all of my tubes are labeled. I have test tubes 1 through 5 and control tubes 1 through 3. So now that we have all of our glassware labeled, all of our microcentrifuge tubes labeled, and we're wearing the proper protective equipment of our gloves, lab coat, and goggles, we're ready to begin our experiment. So now we're going to add the first fluid to all of our test tubes, which is going to be the water. To make sure that we're adding the appropriate amount of each reagent to the tubes, we're going to add all of one reagent to a tube all at the same time. So rather than do everything that needs to go into test tube 1, then everything in test tube 2, we're going to add all of the water, all of the substrate, and then all of the pH buffer, and then all of the enzyme. We're going to start off with water. We're starting with water because we also still need to practice our micropipetting technique. So this here is a micropipetter. It is capable of moving very precise volumes. We like to use these instead of something like a graduated cylinder, especially for moving very small volumes like we're going to be using for these microcentrifuge tubes. The amount of water that we're going to need to add to our first set of microcentrifuge tubes, our controls, is going to be 300 microliters. So I'm going to turn this knob here attached to the plunger until we get to 300 microliters in the viewing frame here. So now that it reads 300, I'm going to go ahead and add the water to our beaker. Now we're going to get the pipette tip onto our micropipetter. To get the tip, we're going to place the end part of the micropipetter into the tip and push down gently until it slides up to the base there. Then we'll close our pipette tip box and put it off to the side. Since we're moving all of the same fluids here, we don't need to use multiple pipette tips. We can use the exact same pipette tip and add it to the solution each time. To use the micropipetter, we're going to depress the plunger, which is this part of the micropipetter here. We're going to push it down. You can push it down twice. There's an initial soft stop and then a second harder stop. When we're drawing up fluid, we only want to push it down to the first stop. So we're going to depress the plunger to the first stop, which is going to expel air from the pipette tip, put it into the solution that we want to draw up, and then slowly release the plunger tip until it's at the top position. As you can see, the water stays within the pipette tip, and so that we can transport it to different places that we need the fluid to be. Then, to expel the fluid, you merely push down on the plunger until you get to the first stop. Now sometimes, I don't know if this will come up on the camera, but you can see that there's a small amount of fluid still within the pipette tip. To get this last bit of fluid, we're going to push it down to the second stop, which is going to expel an extra bit of air to fully void the pipette tip. Then, when we're done with our pipette tips, we're going to dispose of them in the waste container here. There's this button at the top here near the plunger. If we push this down, it'll launch the pipette tip into the pipette waste container. We're going to want to do that every time we need to change pipettes. What you don't want to do is pull or yank on the pipette tip, because if there's any fluid still in there, it might come out and spill on you. So you always want to push the button and void it into the waste. So now we're going to add the fluid to each of our solutions, or each of our test solutions here. So what I'm going to do is I'm going to open up each of these test tubes so that I don't have to be fiddling around with opening them in between the addition of each fluid. And then after I add it, I will close each tube. So we're going to start off by adding the 300 microliters of water that we need to add to our control tubes. So I'm going to get another pipette tip, push down, close the box, put it away. I'm going to check to confirm that the volume that's going to be drawn up is still the 300 microliters that I want. I want to do this quickly before each time I add solution, just to make sure that the volume hasn't changed. I drop my fluid. Now that I've added fluid to this, I'll close it and then move on to the next one. Now, the control tubes have more water in them than our test tubes do. So now I need to lower my volume from 300 down to 150. Now that it's at 150, I can continue. Now that I've added water to all my tubes, I'll now need to dispose of my pipette tip. And I'm ready to move on to the next solution. So now that we've added water to all of our solutions, we're now going to add the pH buffer. So I'm going to open up each of the tubes. And I'm going to get a new pipette tip. I'm going to double check that my volume is still 150 microliters, which it is. So now I'm going to transfer 150 microliters of the pH 5 buffer to each of my tubes. So each time I'm making sure to slowly release the plunger as I'm drawing solution, to slowly depress the plunger down to the second stop when I'm adding the solution to the microcentrifuge tube. Once I'm finished, I'll discard the pipette tip and I'm ready to move on to the next solution. So out of our tubes, the only thing that we need to add to control 1 is going to be the sucrase enzyme, which we're going to add to all of them at the end. So I'm going to go ahead and take this one and put it aside. Control 3 is the only one that's going to get glucose added to it. So I'm going to also set this aside and add the glucose to it now. This one, the control 3 is going to function as our positive control. It's the one that shows us what a positive glucose signal is going to look like on our test strips later on in the experiment. So I'm going to go ahead and open it up. Get another pipette tip. Confirm that my volume is still 150 microliters. Then draw up my glucose solution and add it to the control tube. And dispose of my tip. Now we're ready to add the next solution. Now we're ready to add the sucrose to our remaining tubes. Just as with all the other ones, I'm going to go ahead and open all of the ones that I'm going to be adding sucrase to. We've already added everything that we need to, controls 1 and 3, so we're going to keep those closed and set off to the side as to not confuse them. I'm going to get another pipette tip. Check to make sure that my volume is still set to 150 microliters. And then I will draw up and add the volume to each of my tubes. Sometimes the microcentrifuge tubes can be a little capricious and don't like to close easily. So you'll want to make sure that they're completely closed so that air particles and other things don't go inside them. Then we'll dispose of our pipette tip. And we're ready to set up the final step of the experiment where we add the sucrase and then put them in their incubation baths. So now we're going to add our enzyme to all of our tubes. We're not going to be adding the enzyme to control tubes 2 and 3, so we're going to set these aside. We're going to want to try to do this as quickly as possible so that we can get them into the hot water bath as quickly as possible so that the reaction can continue and start at the proper temperature. So I'm going to go ahead, as I have been doing, is opening each of these so that I can add the solution. We have the invertase off here to the side and ice. I'm going to go ahead and get my pipette tip ready and check my volume, which is still 150 microliters. I'm going to open up the invertase tube. put the excess back in the ice bath, and then discard my pipette tip. Since timing was crucial there, I'm going to wait till the end to close all my microcentrifuge tubes so that I can add the enzyme all at the same time, so that each of these vessels are going to have similar conditions as possible. Now what I'm going to want to do is give them a couple flicks gently, as so, to get all of the fluid that might be on the side of the tubes to condense down to the bottom. You don't want to give more than just a light tapping, otherwise you might spray water up and undo what you're trying to get done. Now we're ready to load them into our floater and place them into the hot water bath. So now we're ready to put it in the hot water bath. Now our test tube 5 isn't actually going to make it into the hot water bath, so we're going to take it out and put it off to the side, and we're going to measure it. Then we're going to put this in to the hot water bath and remove each one of our test tubes every 5 minutes. So now we're going to measure the glucose that's in our first solution, T5. So we'll dunk the strip for a few seconds, then remove it. Then we'll allow it to develop for 30 seconds. We'll want to take the development color immediately at 30 seconds, as it may continue to develop beyond that and give us a false reading. So now that it's developed for 30 seconds, we'll take the color reading now. We want to match it to whichever one appears to be the closest color to the diastick, using the answer strip here. So now we're going to take tube 4 out and measure the amount of glucose present in tube 4. We'll open the tube, put in our diastick dipstick, pull it out, and then wait 30 seconds for it to develop. So even though we can see it's already starting to change colors, we want to make sure we wait the full 30 seconds, giving it the appropriate time to develop. So now it's fully developed and we can record the color and the concentration. So now we're going to record the amount of glucose in test tube 3. So we'll take our diastick's dipstick, place it into the solution, and then allow it to develop for 30 seconds. So now that it's finished developing, we can record the amount of glucose that has been produced. Now we're going to measure the amount of glucose present in our second test tube. Dunk our diastick in. Then we're going to let it develop for 30 seconds. Now that the stick is finished developing, we're going to record the color and the concentration of glucose in our sample. So now we're going to remove our 3 control tubes and our final test tube and measure the glucose present in each of them. We're going to start off here with our test 1 tube. So same process as before. We'll allow the stick to develop for 30 seconds. While it's developing, we'll go ahead and measure our other ones as well. So we'll have control 1. This strip is finished developing. We'll have control 2. And then we'll have control 3. So now this stick is finished developing. And this stick is finished developing. And now control 3 is also finished developing. So feel free to rewind and go back and pause the video at each of the development times to make sure that you get the correct color and concentrations. And then from here we're going to analyze the data that we've collected and move forward in the part 1 of this lab.